A very good morning to all. The title of this video assignment is New Antibiotics Discovered by Sifting Through the Microbiome, and it will be presented by me, Melissa N, and my partner Guinevere. In this video assignment, we will talk about the overview on the discovery of antibiotics, SynBNP approach, discovery of human mycin in vitro and in vivo, and the mode of mechanism of beta-lactam antibiotics. Now, I would like to talk a little on the overview of this topic. This was a recent study that was discovered by researchers at Rockefeller University have found a way around this problem by using computational methods in order to identify which genes in a microbe's genome which are able to produce antibiotic compounds. By testing the compounds against the human pathogens, researchers successfully had identified two closely related antibiotics which are human mycin A and human mycin B. Both of these antibiotics are found in a family bacteria called Rhodococcus. Human mycin has proven the effectiveness of it against Staphylococcus as well as Streptococcus bacteria, which both of these bacteria are able to cause dangerous infections in humans and tend to grow resistant to various antibiotics. Mechanism of human mycin is mainly worked by inhibiting enzyme that bacteria use to build cell walls. And if the cell wall building pathway is interrupted, then the bacteria will initially die off.
SynBNPs, which stands for Synthetic Bioinformatics Natural Products Approach, is used whereby natural product structures are bioinformatically predicted from primary sequence and produced by chemical synthesis. This approach circumvents the need for bacterial culture and gene expression and is applied to non-ribosomal peptide synthetase gene clusters from human-associated bacteria. The SynBNPs predicted is screened for antibacterial activity against common human-associated commensal and pathogenic bacteria, which led to identification of humimycin A and B. What is non-ribosomal peptides? It is one of the most common secondary metabolites produced by bacteria. The humimycins were predicted from closely related NRP genes cluster found in the genomes of Rhodococcus equi and Rhodococcus erythropolis respectively. These two NRP genes cluster encoded heptapeptides that differ at only the 4th and the 7th residues. These figures shows the steps of prediction of natural product structure from primary sequence. Now we are going to proceed to the discovery of humimycins in vitro. The humimycins were broadly active against firmicutes and showed some activity against actinobacteria. It is particularly active against Staphylococcus and Streptococcus, including common human flora such as Staphylococcus aureus, which requires minimum inhibitory concentration of 8 microgram per ml, and Streptococcus pneumonia, 4 microgram per ml. Humimycin A MIC values were 8 to 128 microgram per ml against MRSA clinical isolates. To study the antibacterial mode of action of the humimycins, the researchers selected Staphylococcus aureus USA 300 mutants that could survive on 2.5 times the MIC and sequence the genomes of the resistant mutants. Upon comparison to the parent strain, all mutants contain one non-synonymous mutation in SAV1754, an essential gene in Staphylococcus aureus. Overexpression of SAV1754 conferred resistance to humimycin A, showing inhibition of SAV1754 is a likely mode of action of the humimycins. The gene product is believed to be a homologue of MURJ, a flippase responsible for the translocation of peptidoglycan precursors from the intracellular to extracellular. Merck & Company identified synthetic small molecules inhibitors of SAV1754. The ability of SAV1754 inhibitors to potentiate beta-lactam antibiosis is thought to arise from the fact that both antibiotics target the same essential pathway. These figures shows how humimycins and beta-lactam antibiotics target the peptidoglycan biosynthesis pathway. Humimycin A restores beta-lactam sensitivity to beta-lactam resistant bacteria. For example, the MIC of carbapenicillin decreased from 32 to 1 microgram per ml in the presence of 2 microgram per ml humimycin A against MRSA USA 300. Humimycin alone showed no detectable antibacterial activity.
Now I'm going to talk about the mode of mechanism of beta-lactam antibiotics, which leads to inhibition of cell wall synthesis. The particular group is characterized by its four-membered nitrogen-containing beta-lactam ring at core of their structure. Beta-lactam will target penicillin-binding protein, which is a group of enzymes found in the cell membrane involving the cross-linkage of bacterial cell wall, hence unable to form cell wall, and bacteria will die. The discovery of humomycins in vivo. To explore the ability of humomycin A to potentiate beta-lactam activity in vivo, researchers has infected mice with beta-lactam resistant Staphylococcus aureus, a gram-positive bacteria. The mice treated with mixture containing humomycin A and beta-lactam antibiotic. It shows an effectiveness in inhibiting the synthesis of cell wall. In a mouse peritonitis sepsis model, treatment of a MRSA cold infection with dichlorcecilin and humomycin together dramatically increased survival compared to treatment with either humomycin or dichlorcecilin alone, potentially providing a new MRSA treatment regimen. In a summary, the production of these antibiotics, specifically humomycin A and humomycin B, able to act on against firmicutes and actinobacteria, has been found from a platform known as SynBNP approach that has proven the effectiveness of these antibiotics to be a new treatment regimen in the microbiome by suppressing the synthesis of cell wall, which is essential in bacteria. Hence, these are our reference. Thank you very much.